And when we talk about bioindicators, things that help us to understand if the soil food web is active, is functioning, if everything that we need is there. I wanted to go, because we're presenting this to a group of people that work in Africa, to the Serengeti, and say, there's estimates of like 2 million animals, many different kinds of animals in the Serengeti, at least during the big migration event that happens. And I want to have people think through, Elaine, how would they be able to use animals as bioindicators? What if those populations change? What do you think about that?
Yeah, well, to me, a bioindicator is something that's going to tell me whether things are going right or things are going the wrong way. And there's way more ways to go bad than there are ways to go good. There's always a few, but you're limited in the beneficial direction. So
Mm-hmm.
you've got to pick out something that these organisms do, large or small, to make things better or make things worse. Or in a lot of cases, people would look at just the number of animals that are out there. And if the numbers have decreased significantly, then what's causing that decrease? And I want my bioindicator to help me understand why are these organisms are why would these populations suddenly start dropping.
Mm-hmm.
Where's That's the information I need. Are they being killed off by there's an outbreak of ticks
Mm-hmm.
that are carrying disease to every single one of these animals, and at a certain time, long enough time, these guys will start dropping dead. So
Absolutely.
what's the bioindicator there? Well, isn't it really the ticks? And we
Yeah.
should be assessing how many ticks are out there. It's way easier to do that than sometimes trying to understand why our animals are behaving like they're sick, they're unhappy, they're slow, they, kind of glazed over eyes. Yeah, they're sick, but what's causing that effect? What's wrong with the environment here that this disease-causing organism is capable of coming back at us? That why didn't these animals gain resistance
Mm-hmm.
to the tick or to the organism, the bacteria that was carried by the tick, that was carried by, yeah, the animals?
Mm-hmm.
So you have to work through, you got to keep going further and further back down the chain until you get to something that you can manage to
Right.
get rid of the problem forever. And so
Oh,
most of the time
yeah.
sorry, go ahead.
Oh no, I was just gonna say that's a perfect setup for this, which is, you have over time said, if we focus on these four microorganisms that we can see through a microscope, their populations, their diversity, their abundance is going to tell us a lot about this more hidden world, right? It's easy to see the wildebeest in the Serengeti, and it's more difficult to know what's going on with the nematodes in a soil sample. But it's that concept of a bioindicator.
Yep. And I think it's in a lot of ways it's way more easy to assay for these guys because they won't kick you. You
Yep.
get too close to some of those animals out on the Serengeti, and yeah, you can. It can be a very painful thing, right? So we can look at the wildebeest from some distance, and come back into the laboratory and take a look at the biology that's in your samples, and
Mhm.
this will tell you a lot.
Well, we've got some videos that are gonna bring this whole thing to life. Um, but I just wanted to mention, because you've seen that our audience has seen this graphic before, that we're kind of now really focused on step 4, the monitor the system with microscopy. But really, this step goes into every step that's come before. We have to check with the microscope. Even to start this process of restoration of the soil food web, we've got to know what's missing so that we can go find that thing and get it back into there. And when I say find that thing, it's not like we're going on a hunt for something specific typically, but we're bringing materials from the local environment that is full of this diversity. And then I've also put this arrow here saying we've always got to go forward. Any land management that we do, we need to check. There's a lot of trialing that needs to happen in different environments. Say, does this work, or does this work, or does this work? We need to set those trials up on the farm and then monitor what happens to the soil food web, what happens to these bioindicators.
If the crop I want to grow is pineapple, what do I have to have in the soil to maintain the growth of the pineapple?
Mm-hmm.
Gosh, you need a fair amount of water, maybe less than we think we have. But if we go in understanding that we've got to build that structure, as we were just talking about with the testate amoeba, we've got to build the structure that will hold the water.
Mm-hmm.
And all of a sudden you will be able to grow plants in the dry parts of the desert fine, with no problem, if you get the water right. But you got to stop destroying these organisms in the soil. So it's entirely possible. Let's see if we can repeat what people back in prehistoric times managed to do before we came along with our toxic chemicals and our big machinery that compact and disturb and destroy these organisms.
Absolutely. Well, you mentioned the testate amoeba. We had a great little picture of it, but we also have this video of actually what you might see when looking through the microscope when one of these guys is active. There's a little bottle, and then the organism is coming out.
Now, this has been speeded up by about tenfold. Normally this testate amoeba, and you can see the amoeba in the back. And does my pointer show up, or just yours?
No, it's just going to be mine in this case.
Okay, so so we can see where those pseudopods coming out are searching for the bacteria, and if they come in contact with the bacterium, they're going to engulf it. They're going to come completely around and engulf it into their bodies. If you see that pseudopod starts to go around the bacterium, but decides that bacterium is like, ugh, I don't want to eat that. It will undo the engulfment, just pull back on the pseudopod and go off on another direction. So they're very picky, these organisms. They don't eat just any old bacteria. And which is why we need to have diversity in here, because we want these methods of getting the bacteria, all the different species of the bacteria, get them bringing along the nutrients that have been laid down inside those bacteria. We got to convert them into a form that your plant can utilize.
Absolutely. Well, and I think we have another example here, if I can move forward on the slide, to a fungal feeding nematode that's doing the same thing, releasing nutrients, but rather from bacteria. Mm-hmm. It's looking for some fungi, which I don't think we're going to see it find in this particular field of view.
Yeah. But you can see it searching. You can see where it kind of sticks a little bit of its spear out of its mouth. So that darker kind of crystallized material at the tip of its, we're up there by the mouth. Mm-hmm. You can see that it's perfectly capable of finding a fungal hypha, penetrating the fungal wall, and then sucking out the nutrient content inside that fungal hypha. And then this nematode will just go to the next cell, suck out the real good juicy cytoplasm, inside that fungal cell. And then this nematode moves on to the next one, next one, next one, just right on down the line. And so you can imagine that fungi don't like to see this guy coming.
But plants do, because as it's sucking those juices out, there's a lot of excess nutrients in that cytoplasm that this organism is going to excrete as waste right near the roots of the plant, where the plant can then say, ooh, there's some extra nitrogen. There's some extra zinc.
Yep. Thank you, Mr. Nematode, for bringing me these good things to eat. Because the fungal feeding nematode knows that it's important for it to make certain that those nutrients are released into the soil so that the plant can continue to grow. Mm-hmm. The plant can continue to do photosynthesis, and those carbons come down from the leaves of the plant, going to mix with the nutrients that are being sent its way by the nematode, by this fungal feeding nematode, releasing the too high level of nutrients that are contained in the bacterium or in the fungus. And so that's how the plant gets that soluble nitrogen and can easily take it up into its body. Now, if the plant gets all of the nutrients that it needs and doesn't take up all of the soluble nutrients that were released by this nematode, what's going to happen to those soluble nutrients that are running around in your soil? That isn't it going to cause some damage? Or, why can't you hold, or do you hold up, hold those nutrients in the body somehow, some way? And of course, it is other bacteria and other fungi that can take up those soluble nutrients. If the plant doesn't take it up, these other organisms are going to take up those nutrients so that no leaching loss ever occurs in a healthy soil.
Yeah, that biological reservoir is so powerful as compared to the way that the Green Revolution said, just dump on tons of nitrogen, and then it all ended up in the rivers. It all ended up in the atmosphere as a greenhouse gas. It all it caused horrible problems. Now this one is a little bit more rare, this next slide, but we love to show this one because it's actually two organisms, and it's a rotifer, which is more like a micro animal technically, and a tardigrade, which I think is the same category, or sometimes they're called water bears. But it just kind of looks like this little rotifer, which is this moving organism here, is kind of going along the back of this water bear and cleaning him off. Maybe sucking some extra bacteria off of his surfaces.
Yep. And that water bear or tardigrade is just like, yeah, this is as good as a massage. Oh, yeah, a little more there. Yeah, I'll get rid of those little bacteria. That rotifer is having a grand old time with the feast offered by the tardigrade and doing a service for the tardigrade. Isn't that amazing?
And if we saw a lot of rotifers, we might be concerned that that sample is kind of going low oxygen, right? They tend to thrive in those environments. But the tardigrades, which we don't see that often, or at least I haven't in soil samples, but to me, I get really excited because you talk about how they're taxi cabs that move things around in the soil and help those beneficial organisms to spread.
Yep, and they definitely consume the smaller organisms in the soil. So bacteria and fungi are eaten by the tardigrades and release those excess nutrients into the soil, so your plant can have its pick and choice of what's being offered. They're just so cute. The tardigrades their little feet, and if you ever see one walking across the slide, it's just, they are so cute. You have to follow it. There's just, you have no other choice.
Yeah, I think they should be the unofficial mascot of the soil food web school. And then here is a really interesting slide where I think this was a tea or an extract that somebody, I think it was a tea that Keisha was
Yeah.
brewing. And we actually see just an explosion of ciliate populations, which can indicate to us that we actually have something maybe a little out of whack here, that they don't have enough competition from the other kinds of protozoa in that community, or that they're finding, like, see, they're all jammed up here. Maybe they're finding pockets of really low oxygen where there's some kind of anaerobic bacteria, and that can actually be like a negative
And
indicator, right?
see that big pocket over on the lower left side, right
Mm-hmm.
in there? They're all belly up to the bar, almost like there's got to be something that they're eating in there. They're slowly tearing it apart. And that's what's causing the intense movement of
Mm-hmm.
these ciliates. And they are keeping oxygen levels low
Mm-hmm.
because they're so active. They're using up so much oxygen, chowing down on these couple of pieces of organic matter that's in this sample.
Mm-hmm.
Now, from here, this magnification is not high enough for you
Mm-hmm.
to see the characteristics of ciliate. So please go look at the microscope classes that we offer so
Mm-hmm.
that you can see the characteristics of these ciliates and why they get called ciliates. They have little tiny hairs over a certain portion of their bodies, and all those little hairs are called cilia.
Mm-hmm.
And they work together to propel the cilia zooming around. And you've seen how much they zoom all over the place. And so just classic pictures, and you'll identify them going up one more um, level of magnification on your microscope, and you'll
Mm-hmm.
see all those little hairs. You'll see the very characteristic examples of what they look like close up.